Let's say you work at the Center for Disease Control and you are tasked with analyzing this genetic disease that is known to be dominant recessive. In other words, if a person has both defective alleles, they got one defective allele from mom and one from dad, they will have the genetic disease. And we can think of sickle cell anemia, for instance. And what you want to find out is, based on the number of people that you have documented who have this disease, what percentage of the overall population is a carrier for this disease. And the way you would do that, once you've established that you are working with a complete dominant trait, you can turn to the Hardy Weinberg equation. And the Hardy Weinberg equation has two parts, and what I'm going to do in this video is walk through how we can do it. And to change the tone of this video, instead of looking at diseases, we're going to be looking at flowers. And in this case, we're going to be looking at red and white tulips. And in tulips, we know that the red pigment is dominant to the white pigment, or the absence of pigment. Therefore, if I crossed a red true breeding tulip with a white true breeding tulip, I'm going to get all red tulips as their offspring in the next generation. And so, based on a patch of tulips that I've examined, I've found that 5% of them are white. And the question is, what percentage of the tulips can we estimate with some statistical accuracy are going to be heterozygous, are going to be carriers for that white gene? And so the first part of the Hardy Weinberg equation is very intuitive. So the first thing tells us that P plus Q equals 1. And what this really tells us is that the number of R alleles, big R alleles, plus the number of little r alleles must sum to unity. So if you had 30% big R alleles, you must have 70% little r alleles. The next part of the equation tells us that the genotype ratios must also sum to unity. And what this is P squared plus 2PQ plus Q squared must equal 1. And so what does this mean? We can break this down and think of it as the homozygous dominant, big R, plus big R plus little r big R plus little r squared, or little r. So these are all the possible genotypes you can get, and the reason we call it 2PQ is because it's really, it can be either big R or little r big R, because it ends up with the exact same result. And this must equal 1. So this 5% figure that we've been told must be homozygous recessive, must be white flowers. What this means is that Q squared must be equal to 0.05. And so if we take the square root of both sides of the equation, what we find is that Q, the ratio of recessive alleles in your population, must be equivalent to 0.22. So 22% of all the alleles you're going to find in your patch of tulips is going to have the little r gene, which codes for the absence of pigment being created. And so now that you have Q, you turn to equation 1. And with equation 1, we know that P must be equal to 1 minus Q by simply algebraically rearranging it. And what this tells us is that P, therefore, must be equal to 0.78. In other words, 78% of the alleles in your tulip patch are going to be the dominant allele big R, coding for the red pigment. Now, what we want to do is find how many of the tulips in our field are coding for, or have the heterozygous genotype, that are carriers for the little r allele. And to do that, we're going to turn to the second equation. So now that we've established what little p is, we're going to have 0.78 quantity squared, just plugging and chugging, plus 2 PQ plus, and then 0.22 quantity squared must equal 1. And solving for 2PQ, what we find is that it must be equal to 0.34. And what this figure tells us is that 34% of the tulips are carriers. And so what we saw was, with some very basic statistics and algebra, we were able to look at just one number, just the ratio of the number of homozygous recessive traits or tulips that we found. And from that, we were able to not only deduce the number of carriers for it, but as well, we're able to statistically predict the number of homozygous dominant, because homozygous dominant is 78%. Homodominant. And we will see that these numbers will sum up to unity, which is what we expect. But the key takeaway from the Hardy Weinberg equation is also the fact that we need to make sure that the trait we're looking at is complete dominance and it only involves one allele. If that isn't the case, this thing breaks down and these two simple equations don't work anymore. But for cases like this, and it does apply in nature, like we see with tulips, we can make these very nice estimates using statistics. And so I hope you guys find it useful. Let me know if you have any questions, and thanks for watching.